Thanks for the introduction and the invitation. Today I'm going to talk about universal donor stem cells and its work that was done both at the University of Washington and at a company called Universal Cells, which is about 20 minutes away on First Avenue. So the cell I'm going to talk about is the pluripotent stem cell, either an embryonic stem cell or an induced pluripotent stem cell. And of course, the reason we are interested in these cells is they have incredible therapeutic potential. They can turn into any cell type in the body, in theory at least. And of course, each of those cell types could be used to treat many different diseases. So it really has the potential to transform medicine if we can figure out a way to get these cells into patients and to prevent them from being rejected, which is really the focus of our work. So there's really two ways to go about the rejection problem. In the first, you try and actually match the cell to the recipient. And of course, mainly we're concerned with the HLA molecules. And that's shown here, the HLA matching strategy, where you would have either autologous stem cells, these would be induced pluripotent stem cells made from each patient, or you could create a bank of pluripotent stem cells where you would probably need hundreds of different lines to cover most people in all their different HLA types. But either way, you're talking about multiple derivations of stem cell lines. You're talking about doing all of this in a clinical grade processing unit with tons of paperwork and procedures. You're talking about differentiation protocols on hundreds of different cell lines that could be very unpredictable. And then you're going back into the patient. So it's a very expensive and time-consuming process. Whereas the approach we're taking is to make a universal donor cell. And in this case, we have this master cell bank that we make into a non-immunogenic cell line. And this is done in the undifferentiated state of the stem cell, all of the genetic engineering. At that point, you can then differentiate the cells very predictably because you're always using the exact same cell line. And these cells would be able to treat all individuals. And of course, there are lots of obvious advantages to this approach compared to the autologous. You get this very thoroughly characterized product that you can spend months analyzing before you give it to the patient. You can manufacture at scale, avoid immunosuppressive drugs, which are currently used in a lot of these cell therapy protocols. And most importantly, we think that's going to improve trial design, lead to better patient outcomes because every patient in the trial is getting the same cells, which will allow you to do a lot better job in your clinical studies. So if we want to make a universal donor cell line, we have to deal with HLA, the human leukocyte antigens that are responsible for allogeneic rejection. And there are two types. There's the class 1 molecules, which are composed of these variable heavy chains, HLA-A, B, and C, that are highly polymorphic, and a common beta-2 microglobulin subunit. And these HLA class 1 molecules are expressed on almost all cells in the body, and they present peptide antigens to CD8 cells. Class 2 molecules are another story. They're two different variable chains. These are DP, DR, DQ. They're mainly expressed on specialized antigen-presenting cells, and they present peptide antigens to CD4 positive T cells. So we have to deal with these HLA molecules if we want to avoid rejection. And this is not a new concept. It's almost 30 years since the original beta-2 microglobulin knockout mice were made by two groups. They were one of the first knockout mice ever made. And of course, if we knock out beta-2 microglobulin, what these papers showed was that none of those class 1 molecules get to the cell surface. All of the HLA-A, B, and C chains are still expressed in the cell, but they don't get to the surface. So you have no class 1 molecules on your cell. And this, in many ways, was sort of a first-generation universal donor cell. These mice that were beta-2 knockouts, people could take organs and cells from a beta-2 knockout mouse and show that they would persist longer in an allogeneic strain of mice. So these were these sorts of universal donor transplant experience with organs, and that was done with kidneys and islet cells. Livers and hearts. So a lot of data suggested that this might be a reasonable approach to making a universal donor line. But of course, it was soon shown, very soon after the mice were made, it was shown that it really wasn't that simple. And in fact, cells that have no class 1 molecules on their surface tend to be rapidly destroyed by natural killer cells. And that was shown in this paper in '91. It's called the missing self hypothesis. So the idea is that it would be a bad idea if your cells eliminated all of those HLA molecules. You'd have no way to recognize any sort of foreign peptides, and we would want to eliminate a cell like that. And the NK cells take care of that job. This is the missing self hypothesis. It's caused by a lack of binding to inhibitory receptors on the NK cells, which recognize all those class 1 molecules. And it gets pretty complicated when you look at all the receptors involved. So that complicates the process. If we want to make a universal donor line, we're going to have to deal with this NK cell issue. So the way we approached it is to not just remove beta 2 from cells and get rid of all class 1 molecules, but rather to make a fusion protein where we take the beta 2 subunit and we fuse it to a heavy chain to make what's called a single chain HLA molecule. And in this case, rather than a polymorphic heavy chain like HLA-A, B, and C, we use a non-polymorphic heavy chain, and we're using the molecule called HLA-E. So everyone has the same version of this non-polymorphic cell, and it won't be recognized as foreign. And the molecule, as I mentioned, that we chose for this is HLA-E. Here's a picture of it. It's like another class 1 molecule. It presents peptides. It has a beta 2 subunit. It's called a non-classical class 1 protein. It's not polymorphic, as I said, so we won't have rejection. Interestingly, it presents peptide antigens that are the signal sequences of other HLA proteins, which is why it's really the center of this missing self hypothesis, because it's actually telling you whether or not the HLA is expressed by putting these signal sequences as peptides on the surface. And it's been shown in many different tumor lines that it can block NK-mediated lysis, which is what we want to do. We know the receptor that it binds to, this NKG2A receptor that's present on most NK cells. So if we can make this molecule, we think we can block that missing self phenomenon. And there's actually a similar molecule called HLA-G that works similarly in the placenta, which would have been another option. But I think HLA-E is a better place to begin. So how are we going to do this engineering? Well, we need to do gene editing, and we need to do it in a way that's clinically acceptable if we want to use these cells in people. And we use the technology that I worked on in my lab for many years at UW based on adeno-associated viruses. These are parvoviruses. They've been crystallized. They have a single-stranded linear genome with these invertible repeats at the end, and we just replace the guts of the virus with whatever our targeting construct is. Importantly, this is nuclease-free. There's no CRISPR involved. There's no genotoxicity, no off-target cutting, no on-target mistakes. It's very simple to deliver. We package it in this particular serotype that efficiently infects stem cells, and we just add it to the media. We don't have to electroporate or lipoffect or any of those toxic treatments. And finally, AAV vectors are in the clinic already. There's not going to be any regulatory issues about using these cells, these vectors. So you might ask, well, if there's no nuclease, how do you do gene editing? And that's a fair question. So this is how it works. It pairs with actually the replication fork. And as some of you may remember, you have your leading strand and lagging strand at the fork. And because of the direction of replication, the lagging strand always has this little single-stranded gap before an Okazaki fragment can form. And that's where the AAV pairs at the replication fork, at the lagging strand, and we've shown that experimentally. So it actually looks kind of like an Okazaki fragment. You can trim off these ITRs with various cellular functions, and it will get incorporated into the lagging strand just as an Okazaki fragment would, along with whatever the mutation you're trying to introduce. And then you can do another round of DNA synthesis or repair synthesis to get the second strand edited. And that is how you can do the editing without a nuclease, without breaking the DNA. So we designed several vectors to go after beta-2 microglobulin first and deal with the class 1 story. a couple of these vectors are simply knocking out beta 2, and we insert a selectable marker, in this case a high-tech gene or a TK-neo gene. And we have other vectors that are going to knock in our single-chain protein. So in this case, we have a beta 2 microglobulin reading frame, so we need to knock in that HLA-E reading frame to make a single-chain protein molecule. And we made two versions. One of them is a dimer, and that's shown here, where you have the beta 2 fused to the HLA-E heavy chain, and you would present whatever peptides are normally presented by HLA-E. And as I mentioned, these are mainly signal sequences of other HLA molecules. And we also made a trimer, where you can actually have another covalently attached peptide and put in a specific peptide that you want to present. So we made that. In this case, it's the peptide from HLA-G, which is non-polymorphic, and we thought should work. So we had 4 different vectors that we made to try and engineer cell lines. We started off with pluripotent stem cells, in this case an embryonic stem cell line, and just started doing the editing. First step, we knock out the first allele of beta 2 microglobulin, get about 10% of our selected cells are edited. We then either knock in the dimer or the trimer molecules in a second step, and in the case of the dimer, we have to eventually cree out the selectable marker to get good expression. So we have this intermediate that expresses very little class 1. Or we can just simply knock out both alleles of beta 2 and get a totally beta 2 negative cell as a control, which we did as well, although I'll point out that actually these cells don't grow well. Turns out that the human stem cells don't want to be beta 2 negative, and they tend to become abnormal. So for some of our experiments, we use this as a class 1 negative line. And then we can show by flow that the cells are expressing what we think. If you look at a wild type cell, you can see the orange tracing is adding interferon gamma. The red is the isotope, and the blue is no interferon gamma. You can see that you get interferon gamma inducible expression of E, A, B, and C on normal wild type cells, as you would expect. If you make a beta 2 knockout, none of those molecules are on the surface. If you express the dimer, you can see that E is in fact expressed at higher levels than the wild type, and it's inducible by interferon, which is part of the advantage of putting it at that beta 2 locus. It's going to be regulated like the beta 2 gene, like a normal class 1 molecule. And we don't make any A, B, or C. And similarly, we get the same results with the trimer, except we also can detect some expression without even adding interferon gamma. And that's probably because that peptide is stabilizing the protein on the cell surface, making it last longer. So we have what we want. We have a cell line whose only class 1 molecule is HLA-E on the surface. And we can start doing experiments. So the first thing is to see if we can prevent lysis, prevent that missing self response. And what we do here first is we take the pluripotent cells and we differentiate them into CD45 positive hematopoietic cells, where we can do these assays really nicely. And that's about a 3-week process to start from pluripotent to get to hematopoietic. And we can show by flow that the cells express class 1 as predicted. In this case, we use this pre-Cree version as our class 1 negative because the true beta 2 knockout actually doesn't differentiate. And we can do simple experiments, simple immunology experiments to see if it's working. The first one is a chromium release assay. So in this case, we take normal human NK cells from a normal donor, and most of them express NKG2A, which is the receptor for HLA-E, and that's pretty typical. And we label the hematopoietic cells that we engineered with chromium, and then we look for chromium release, and that's how you measure specific lysis. So if we take the wild type cells, we get this much lysis when we add the NK cells. If we take our class 1 negative cell line, we get much more lysis, and that's the missing self response in action in a human cell. In this case, without class 1, you get a lot more lysis by NK cells, and we can show that expressing the dimer of HLA-E or the trimer in an otherwise negative line actually prevents the lysis and makes it last longer even than a wild type cell would last, presumably because there's more HLA-E than you find on a wild type. So we can eliminate that missing self response. We can show with blocking antibodies that it's due to HLA-E binding to NKG2A, as we would expect. And just as an aside, this is great, but it's probably not the total solution. Everything gets better over time. And some donors have relatively low levels of NKG2A. So in this case, we get a partial improvement, but there's some NKs that aren't going to recognize HLA-E, which is something we need to still work on in the future. We can show in vivo that it works as well. And here what we've done is take an NK cell line called NK92. We first label our pluripotent cells with a luciferase marker. We then inject them subcutaneously to get teratomas, which is what these cells normally form in an immunocompromised mouse. And we inject NK92 cells into the peritoneum. And what we can see with luciferase labeling is that when you add the NK92 cells, you see that the class 1 negative teratomas are smaller than the ones that have E, and that's not really the case when you don't add the NK92 cells, so you can detect this NK-specific lysis in vivo and see that you get protection when you express HLA-E. You get even more dramatic results when you first differentiate these cells into hematopoietic cells and then inject them, and follow that with an inject these cells and follow that with an injection of NK92 cells. Before you inject the NK92, you see nice labeling of the whole peritoneum. And afterwards, what you can see is the class 1 negative cells are completely eliminated by the NK92 injection, whereas the ones expressing HLA-E are not. So we can really nicely demonstrate protection in that model. What about T cells? So I focused on NK cells. That's the missing self-response. But of course, T cells are a major component of rejection. And what we would predict is that because we've removed class 1 from the surface of the cell, that no CD8 positive cells should recognize and kill our cell line. And the way we showed that was to first take normal human CD8 positive cells. We prime them on the wild type line that is expressing all those HLA antigens, so we get CD8 cells that are highly reactive against those HLA molecules. And then we can do a chromium release assay, similar to what I showed you with the NK cells in vitro, show that the wild type cells are rapidly lysed by these CD8 cells, whereas none of the engineered lines are, including when they express the dimer or trimer. So it's not recognized by CD8 cells, which is what we expected because it's not polymorphic and it should be recognized as host. And then we can do similar in vivo experiments as well. In this case, we labeled these CD8 cells with a red dye and saw how they bound to the teratomas. And you can see preferential binding to the wild type teratomas in comparison to the negative E class 1 engineered teratomas. And you can quantitate and show that it's significant, show that the tumors grow better with the HLA-E expressed, and find the CD8 cells on histology. And of course, the third component of the immune response is the antibody response, which actually plays a pretty big role in rejecting allogeneic grafts. And here what we've done is take human sera from patients that had received transplants and developed anti-HLA antibodies, and we can show that our cells also do not react with those in an antibody-dependent cellular cytotoxicity assay. So the wild type cells are lysed by the appropriate sera that actually react with the known HLA molecules on the surface of these cells, and that doesn't happen in the knockout or the E trimer line. This is probably an artifact as it's a polyclonal sera. So we can show basically that these cells avoid avoid rejection from T cells, NK cells, and also B cells that make the antibodies, which is pretty much the whole immune system. We can turn these cells into other things besides hematopoietic cells. This is a collaboration with Dennis Clegg, where we made RPE, retinal pigmented epithelial cells, out of our cell lines, and also showed that they're not rejected by allogeneic T cells. That's one of the first therapeutic uses of pluripotent cells, are these RPE cells to treat macular degeneration. Okay, what about class 2? A few minutes on class 2. It's expressed only on mainly on antigen-presenting cells like dendritic cells, B cells, macrophages, but also it may be expressed on a lot of other cell types in the presence of inflammation. And there's a lot of data to suggest that it may be important there too. So we think we need to get rid of class 2. Class 2, unlike beta 2, unlike class 1, there's 2 variable chains. There's no common subunit. There's no simple knockout strategy like with beta 2. But there are a group of patients that have a disease called Bayer lymphocyte syndrome that lack class II expression on their cells. They have mutations in all these transcription factors that turn on all the class II genes. So our strategy, rather than going after all 6 class II genes, we go after one of these transcription factors. And we chose RFX A and K, which is what most of the patients have mutations in and most was known about. And we can show that when we knock out both alleles of this gene with AAV, once again, the same gene editing process, turn them into hematopoietic cells. They still make hematopoietic cells that are 45 positive, but they're no longer class II positive. This is with HLA-DR. And we can actually do more significant flow as well with these cells and look at the 3 major class IIs, DP, DR, DQ, and also show that when we knock out RIF-SENC, we get very little expression of those class II molecules. So that's our class II strategy. And there's no missing cell phenomenon for class II. We don't have to worry that they'll be destroyed because they're class II negative. So we just have to eliminate it altogether. And this kind of summarizes in the simple way what we're trying to do. The parental cells can express 6 polymorphic class 1s, 6 polymorphic class 2s, as well as HLA-E. This is a wild-type human cell. We can deal with class 1 by making the beta-2 knockout and a HLA-E knock-in to get cells that only express HLA-E or class 2. And at the same time, I didn't highlight it, but we're also putting in suicide genes. And in this case, we put in a thymidine kinase gene. We think that's important, as I'll mention in a minute. We want to be able to get rid of these cells if they go bad. And then on a class 2, we would knock out 2 alleles of RFX A and K, and also be able to put in additional suicide genes at the same time. And this would give us a cell that is what we call a universal donor cell that only expresses HLA-E as its sole class 1 or class 2 HLA molecule. And you can see that there's quite a number of editing steps to get to this end product. And this is a question that often comes up when I present this story, which is, can you really do like 8 editing steps in a row and still get a normal cell at the end? And I think in the case of pluripotent cells, the answer is yes. And here's an example of where while we were working out this process, we ended up having to do, we did it inefficiently, and we ended up having to do 1, 2, 3, 4, 5, 6, 7 steps to get the final cell line. You can actually do it in 5, but we did it in 7 this time. And even these cells, because the pluripotent cells are immortal, they'll grow forever. There's no problem with senescence. We did whole genome analysis as well as all the oncogene analysis and showed no oncogene mutations, normal karyotype, normal copy number analysis, and normal differentiation. So as far as we can tell, these cells are normal and very similar to the parent. So yes, you can do a lot of editing steps, but you got to check. At each step we check. We do karyotypes and we look at oncogenes, and about 10% of the time we get an abnormality. We throw that clone out and stick with the good ones. So I will just wrap up with some of the ideas about these cells, and these are probably questions some of you might have. What about tumors? If we don't have HLA on the surface of our cells, won't it be harder to reject a tumor cell that might grow in the body? And that is certainly an issue that we're very concerned with. First off, we know from mouse experiments, as well as some of these people that have known mutations in class 1 and class 2 molecules, that they're not tumor prone. So you're not more likely to get a tumor if you lack these cells. But that doesn't deal with what can you get rid of a tumor if it does form. As I mentioned, we put multiple suicide genes in our cell line, and actually the final product will probably have 3 or 4 suicide genes. So you'd have to get 4 independent mutations to prevent those suicide genes from working. And you need that with suicide genes. You can't have just one, because 1 in a million cells will be mutant in that gene, and you'll rapidly lose protection. So you really need 2 alleles of 2 different genes if you want to be safe, and that's our goal in the engineering process, is to be able to eliminate the genes if we can, if they go bad. Now, if you're thinking through, that may not always be good. If you end up replacing somebody's heart with pluripotent stem cell derived cardiomyocytes and they get a tumor, You might end up getting rid of some of the normal cells too, and you wouldn't want to do that if your heart was composed of those cells. So that's why we want two types. Some might kill only dividing cells, some might kill all cells. Depending on the indication, you can imagine you might take a different approach. Of course, a lot of applications are going to involve terminally differentiated cells that are not likely to form tumor, like red cells or cardiomyocytes, in fact, RPE. And we think a major advantage of our approach is that we have one cell line for everyone. We make these very large batches that can be thoroughly characterized, thoroughly sequenced. You can do all your tumor assays in animals. That actually makes them safer than autologous cells that we really don't have the time or money to do that kind of extensive testing on. And of course, a lot of these risks are going to be present for unengineered stem cell-derived therapies as well, not just for universal donors. And what about infection? Same way, a lot of these molecules are involved in presenting peptides from pathogens to stimulate an immune response. So that's another possible problem if we give universal donor cells. I think this is a relatively minor problem. We know from a lot of the mouse models that they can still fight off infections with redundant mechanisms. And of course, in the case of treating humans, we're going to probably be treating humans that have a normal immune system with a small bunch of transplanted cells in one location of the body. So they're not completely immunocompromised themselves. They have a normal immune system. You will not have to immunosuppress them because the cells don't get rejected. And you could also transplant the cells internally where they may not get exposed to a lot of viruses. And of course, once again, you have this suicide gene option if you need it. So to sum up, what are the advantages? One cell line can treat everyone. I think people get that. You save a lot of time and money and enroll more patients in clinical trials. We think it's going to make a big difference in trying to get pluripotent-derived therapies into people. We can avoid immunosuppressive drug regimens, bank our products ahead of time. That's a big deal. If you're making autologous lines, you start making the line after the person has their heart attack. It can take up to a year to get a therapeutic product starting at that point. So you really can't treat a lot of diseases without having something banked ahead of time. Very important, if you've ever worked with these cells, is you optimize all your protocols for one cell line. And in fact, every ES cell line has its own quirks. And if you want to make a cell type derived from them, you can spend years optimizing a protocol that really only works on that one line. This is an interesting concept, is that the autoantigens and neoantigens are not presented. A lot of genetic diseases, you may never have been exposed to a particular protein because you have mutations in that gene. Then when you get that protein, you're going to have an immune response and reject the cell. That's not going to happen in our cells because they're not going to present the peptide antigens of any protein. So you can do that with both proteins that are mutated in the patient as well as entirely new proteins. You can finally express a bacterial protein in the cell and not worry about rejection. Or you can not worry about autoantigens. If you want to cure diabetes and you give back islet cells, that even if they're autologous, they're going to be rejected, which is why you got diabetes in the first place, because you have antibodies against your islet cells. If we give them universal donor islet cells, they won't present those antigens, and you could actually potentially treat diabetes and not have a recurrence due to the autoimmunity. And finally, I think one of the most exciting parts of this approach is that you make the cell line that you can give to everyone. You have time to engineer it as you want because you only have to do it once. So all additional genetic engineering steps can be added into the system to improve differentiation, to enhance cell function. It doesn't have to be a normal cell. It has to be a cell that cures you and also prevent transformation. So like an enhanced cell function, if you're imagining giving someone T cells to fight off a tumor, you could make a T cell that's more cytotoxic than normal in this case and use that as your therapy. So there's a lot of future science that can be used in this system. So I will stop, and I want to thank especially 3 postdocs from my UW lab who worked on different aspects of this project, as well as our collaborators here, and the group at Universal Cells, which is doing basically the same work in a clinical grade, so that when we get to the end, we'll have a cell line that can be used in clinical trials. And of course, all the funding sources over the years. Thank you.
Okay, we'll take a few questions for David. If you have a question, just go ahead and raise your hand, and we'll get a mic to you.
David, it's a great talk, as always. Very cool work and very exciting. So I guess one question that occurred to me is, do you think that cells, if you had a patient who was transplanted with a universal cell, is there a risk that those cells or their derivatives could actually be transferred to somebody else, and they could actually get be infected with that cell, and if that cell was carrying something, it would also bring it along? I mean, are you creating a vector?
Yeah, so you probably know more than you let on in that question, because there's Tasmanian devils that have transmissible tumors. There's dogs that have transmissible tumors. There's clams that have transmissible tumors that are exactly what he's describing, where it's kind of its own organism, because in the case of the dog, it's been out there for thousands of years, transferring from one animal to another and evolving on its own. It's really sort of like an organism from a sci-fi movie. It's not really a dog anymore. And this is still going on, and people have sequenced the genomes of these dog tumors and shown that they actually have a lot of the same mutations we're engineering. Like, they have an RFXAP mutation, which is almost identical to our RFXA and K mutation. They have a TAP1 mutation, which is very similar to a beta-2 microglobulin mutation. So yes, I'm very worried about universal tumor cells. I wouldn't want to have that be a legacy to leave behind. Although some people argue with me about that. Wouldn't we be better off without people? I mean, come on. But yeah, thus the suicide genes is really critical for that. And also, it's worth pointing out that you'd have to, how would it be transmitted? In the case of the Tasmanian devil, it's by biting. So as long as people don't bite, it might be okay. But in the case of the dog, it's also by sexual transmission. So you could easily imagine someone who got the therapy having sex again. And it's an interesting topic. I could go on for hours about this. But yes, we should think about this. And people often don't realize you can have an infectious tumor cell.